All right. Wow, we got folks from all over today. Got from Canada, California, New York. Great to have you. So let's go ahead and get started. It is 12 o'clock. Thank you so much for joining us today. This is the last of 3 webinars we are hosting this fall. We'll be announcing our 2022 webinar series soon. So keep a lookout for an announcement in our Go Botanical. Or you can check back at our events page at ntbg.org/events, and I'll be dropping those links into the chat box shortly. If you want to be kept in the know and aren't already subscribed to our monthly science and conservation newsletter, you can sign up on our website, and I'll drop that link into the chat shortly as well. So this webinar is being recorded. We'll be sending the recording out to you in the next few days. If you have any questions throughout the presentations, you're welcome to drop them into the chat box. And we'll also be holding a general Q&A at the very end as well. If we don't get to your question, please feel free to follow up with us via email, which I will drop into the chat as well. So we are hosting today's session from our Botanical Research Center on Kauai. I have with me Dr. Nina Ronstedt, our Director of Science and Conservation, who will now tell you a bit about today's session.
Aloha and welcome to this webinar. We're so excited that you're all joining us today. And with this series of webinars we created especially under COVID circumstances, we wanted to invite you behind the scenes so you have a chance to meet some of the people who are leading NTBG's science and conservation work. The flora of Hawaii is really unique, and most of the species found here occur nowhere else on Earth, and this is due to millions of years of isolation. However, our native plants and animals are threatened with more than 130 plant species already known to be extinct in the last 175 years. So that is dire news, but luckily there's a lot more awareness of this challenge today, and we are working with many partners across the state to help stop these extinctions and restore our native forests. And this series of webinars is zooming in on the many different aspects of this work. It's often said that we can't conserve what we don't know, and today we want to celebrate the modern plant explorers who track difficult and remote terrain by foot, rappelling, or with drones to survey and discover new species and populations. Today we'll hear from NTBG's research and field biologist Ken Wood and our GIS and drone program manager Ben Nyberg what some of their most exciting discoveries are, how they found them, and what it means for plant conservation in Hawaii. Welcome.
Thank you very much, Nina. And I think I'll begin. My name is Kenny Wood. I've been with the garden probably since before many of you were born, started around 1982. And I focused particularly on the distribution of rare plants throughout Hawaii. But I've done a lot of work in the South Pacific, in the French Polynesia, Marquesas, Society Islands, the Austral Islands to the south there, and a little bit in Samoa, Micronesia. But since I live here on the island of Kauai, most of my work has been in Hawaii. Now, and one of the hats I wear is doing the field work. I also do a little bit of taxonomic research, and so one of the main aspects of my work includes collecting herbarium specimens, doing the field work. And that's a very important aspect to it. So I'm sent to different places. I've been on many, many thousands of research trips over the last 40 years. And we do have others at the NTBG besides myself. So when I'm talking about this type of work, it's not only me, but there's another great group of researchers here at the garden that have also done this type of work. Now, we've contributed, like I said, many thousands of herbarium specimens, but we also focus on where rare plants occur. And so that means that we need to get out there into the wild to discover these, and that includes a lot of prep work. So a field botanist is going to go organize his field equipment, make sure he has all the proper rain gear and food to survive, and And then we'll either, if we're working in the South Pacific, we usually don't have access to helicopters, but here in the Hawaiian Islands, we've been able to utilize all types of transportation to get to these remote areas. It sounds very romantic and to be an exciting and great profession to be working in the wilds, but there is downsides to it. I remember many winters going out week after week in the rain, camping for 2 or 3 days, getting soaked to death and trying to use newspaper to mount specimens and organize them. And another aspect is that when we get to certain places that haven't been well explored, like the South Pacific, we'll want to get to the outer reaches, the summits, these far, long journeys up very steep areas to be able to document the plants up there. So we have to choose how much food we're going to bring, how much water, how much protective clothing we'll have, because when we come back, we're going to be taking back with us maybe close to 100 different plant specimens and in duplicate for other herbaria. So so there's certain challenges dealing with the weather and the rigorousness of this and steepness of the terrain. But just overall, a lot of it focuses on your familiarity with the species that are out there. So I need to have a background in what type of flora occurs in the region where I am. I'll go out there and I'll be seeing particular types of plant communities. And in those plant communities, I'll be looking for rare species. I'll be photographing them. I'll be making an herbarium specimen if there's enough material for that. I'll also be taking a lot of notes in my field books, talking about the stature of the plant, what the color of the flowers were, is there a scent at that time. I'll also separate a little bit of leaf material from the plant and put it in silica to dry it slowly to preserve the DNA within that plant. And then we do have a DNA library here, a walk-in freezer where we keep our material for future phylogenetic research. So I don't want to spend too much time, and I do, we will have discussions, and I enjoy much more having dialogue with people than monologue, which I'm doing right now. One important aspect also is needing to, when you have a relationship with natural areas, that I think there needs to be a level of respect, and that's what us at the NTBG try to follow. We get our permits through the different agencies, whether they're land managers, the state or the federal government, or if we're working in the South Pacific, we'll go to the local communities and make sure that we have their permission to go up and make these collections, and then their supervising governments, we have all our permits in order for that. So there's a level of respect, and we follow all of the given protocols for doing this type of research. And then also, once we get out of the field after spending 2 or 3 days in extreme conditions, we need to press our plants a little bit better than what we're doing in the field. So we'll be going through all our material, staying up until we get it done, because we're only in these regions for a short period of time and we'll need to get back out there again. So it is a fast moving pace. It isn't as romantic as you would expect it to be, but for me, it's something that I've always loved to do. I never feel that I'm working. It always feels more like I'm in a relationship with the natural ecosystems, and also to highlight it all. It seems that there's always something really unusual that comes out of each trip that we go out and explore. We come back with something really interesting to share with the scientific community, or to bring seeds back, which is another really important aspect to this, that we're collecting seeds and bringing them back and having them grown through horticulture, and also having the seeds stored for long-term preservation. And like I said, I do look forward to having more discussion at the end of this, but Amanda, if you would just go ahead and begin showing just some highlights of some of the work we've done over the last 30 years, and then there'll be a point where we'll talk about just the recent discoveries that were made in the last year or so. Now, is there a soundtrack with this one, or
the necessity for baseline knowledge, guidelines for
there we go.
Sorry, folks. We see. I think the audio and the video isn't on track. We could hear it there for a second, and now we can't
Okay.
again. Yeah, 1 second.
Is that an issue?
Yeah, we can't hear the audio.
Okay, well, I'll just take off my speakers, then maybe I try again now.
guidelines for biotic surveys and
We
inventory.
can hear it. Thanks, Nina.
Necessity for baseline knowledge of species-level biodiversity as a foundation for numerous other scientific disciplines, especially conservation biology. Nevertheless, the field inventory of plants and other life forms is far from complete, and the biodiversity of many tropical areas is rapidly disappearing before it has been adequately surveyed. Advancing knowledge of species distribution and abundance to help prevent extinctions and give guidance towards the management of complex biotic ecosystems has been the fundamental thesis behind NTBG's in situ, ex situ plant conservation research. Throughout Oceania, our floristic inventories have contributed tens of thousands of herbarium collections for scientific research, which include the discovery of around 105 species new to science, and in Hawaii, the rediscovery of around 37 taxa previously thought extinct. Several decades of intense, rugged field research has culminated in the recently published 2-volume Flora of the Marquesas Islands, produced by NTBG and the Smithsonian Institution in collaboration with the French Polynesian Delegation of Research. This research includes the description of over 40 species new to science and is a great example of NTBG's ongoing contributions. A few other examples of NTBG's research and discoveries include our work with the alula, or Berkhamia, a native lobelioid restricted to sea cliffs on Molokai and Kauai, which are in need of hand pollination as a result of the loss of its pollinators. Another is the discovery and conservation of Kanaloa kaholavensis, or Palu Palu o Kanaloa, which means the tenderness of Kanaloa. It was found on a remote dry sea stack on Ko'olawe and is now gone extinct in the wild. Yet it survives in cultivation under the genius of several Maui Nui horticulturalists. The discovery and description of Poliscius flinnii, formerly a Tetrapalsandra, just one of the many new species found in Kalalau Valley, along with Kauai's rarest Kamaka'ala, Geniostoma lawrenceanum, known only from 4 wild individuals, 3 males and 1 female, and now being grown and protected in situ natural areas in partnership with the Hawaii State Division of Forestry and Wildlife. The discovery and description of the gloriously bizarre Sertandra paliku, only known from the cliffs of Keikawiki, Kauai. The region of Keikawiki has always drawn our attention, and when we finally got there in 1993, we were rewarded with finding this Sertandra with stems that looked like the hairy legs of a tarantula. Another interesting discovery by NTBG was the largest representative of Hawaiian Kumu, Doriopteris angelica. Ferns are an important part of the Hawaiian ecosystems, and NTBG now has a fern lab successfully cultivating them. In Hawaii, Melicope, a genus in the citrus family, totals 50 endemic species, making it the most species-rich radiation of woody plants throughout the archipelago. The discovery and description of Melicope stonii in the mesic forests of Kokee was a highlight for NTBG researchers. An example of a plant believed to have gone extinct without successful cultivation is the Kalalau hau kuahivi, Hibiscadelphus woodii, which was discovered in the early 1990s, where there were only 4 individuals on a remote vertical cliff. Unfortunately, after years of monitoring and attempts at cultivation, the population died as a result of rock slides. Today, Ben will discuss its rediscovery with drone research. Some of the more recent work being conducted over the last few years by NTBG includes producing phytogeographical data on the rare thalipteroid fern Menisciopsis wailele. Formerly in the genus Cristella, we are now producing a manuscript for the American Fern Journal. This species was described by NTBG's herbarium curator, Tim Flynn. An important recent finding includes the rediscovery of Cyania cuyaba. This lobeloid was originally known only from Limahuli, Kauai, but went extinct after the 1992 Hurricane Iniki without getting established in cultivation. Now, after its rediscovery, we have been given another chance to prevent its extinction. We currently have a multitude of seedlings and young plants being grown by NTBG and Lyon's micropropagation lab, in addition to several thousand seeds collected over the last few years and being stored in NTBG's seed storage lab. Outplants are also being grown in Upper Limahuli Preserve. Some other good news being made during the summer of 2021 includes NTBG's partnering with the Plant Extinction Prevention Program to finally get successful seed collections of 2 very rare lobeliads, Cyania undulata and Cyania remii, both known from less than 50 individuals. Originally discovered in 1909 and only documented one other time in 1960, the recent rediscovery of Melicope Neale in Kokee, Kauai now gives us the opportunity to prevent it from extinction and learn more about its breeding system and habitat preferences. Just last month, we rediscovered Kauai's Euphorbia Remii variety Layensis after not being documented since 1865. This species in the Euphorbia family, which Hawaiians called a koko, grows into a large tree in wet habitat. A group of around 100 trees were found, and the rediscovery now makes it possible to conserve the species. As natural habitats are changing quickly and plants are retreating to regions very difficult to access, new technologies are being utilized for finding and collecting the rarest species in Hawaii.
All right. Thank you so much, Ken, for preparing that presentation for us. We did have a question come in. And if anybody else has any questions for Ken, please feel free to drop them into the chat or the QA section. So Carrie is curious, what was your favorite or most magical field moment?
Oh, that's a tough question. I think when I first saw the Kanaloa on the sea stack of Aleale, and the way that happened was I had to rappel down the broken cliffs of the main Koholave Island, a very crumbly and kind of dangerous. I had hiked down to as low as I could get to and tied into a cuyaba tree, because in the distance I could see the sea stack and it had a intact shrubland on it. with binoculars, I could see quite clearly that it was intact and no one had documented there. So I did the rappel. I got to what was like a talus connection to the sea stack, and I was able to work my way over and climb onto the sea stack, but it was quite dangerous to get up there. And all the time I'm doing that, it was the first day of spring, March 21st, 1992. There was a mother whale right in Kamohio Bay, the little embayment right there, with a baby whale, and they were splashing the whole time I rappelled down. And there's more to the story. I didn't have enough line when I first rappelled. I had to go back up and then tie a little bit lower, and then got down there and then pull out something out of my vest just to go a little further. But then I climbed up on the sea stack, to make a long story short. And then I saw the Kanaloa, and time kind of stopped still. I could hear the whales in the background and the birds all crying in the sky. And I felt it was just an out of the body experience, really. I was no longer, Kenny Wood. I was just witnessing something, because I knew this. I didn't recognize it as a genus in Hawaii. I knew it was native, and then the story goes on from there. But that was the most magical, or one of the most magical moments. I'm glad I got to share that.
Thank you. Could you tell us a little bit more about the island?
Well, yeah, Koholave was, a military base for many years, and it was being bombed from the air and from ship. There were cattle, there was sheep, goat. So pretty much the main part of the island of Koholave had seen a lot of devastation because of this kind of abuse. So we were sent over to do an inventory, and the top of Koholave, not very high, 900 and something feet, I think, was all hard pan. But there were, like I said, certain little offshore sea stacks and islets that still retained integrity. But Koholave is one of the main Hawaiian islands that few people get to go to, and is now managed by the Koholave Island Reserve Commission.
Thank you. So we had a couple more questions come in. So we have a question from Volker. How do you determine when to bring a rare plant into your nursery for propagation and conservation?
Okay, well, nice question. If I've never made a conservation collection of such a plant, then I would want to do that with the one that was found, because it does have different genetic makeup than other plants. Just like people are different, each one is different and has different genetic makeup. If we don't have enough representatives in a community, there's a problem with inbreeding, and you lose reproductive vigor, and your health isn't quite as good. Same with plants. So when you see rare plants in the wild, there needs to be a lot of discernment and discretion when collecting. But I never take all the seeds, or very rarely do that. And I very rarely take a plant out. It's usually from a cutting, but most often seeds. And then I'll bring those seeds back, and we'll try to grow a certain amount. And then also we'll try to store a certain amount of seed. But basically, it's important to get at least 50 representatives of a species to try to maintain a broad spectrum of their genetic possibilities.
Thank you. CK says, plants are retreating, gave me a funny image, but how do they really relocate to more hospitable niches? And then dash birds.
Okay, yeah, well, so, it's kind of like my hairline. They are receding. Mostly it's not that they're running, because plants, once they're rooted, they're pretty much there. But they're retreating to areas where there's less traffic. There's less goats chewing them day in and day out, or less people driving four-wheel vehicles over them, or certain bases that I surveyed throughout the Pacific. Sometimes they'll do ordnance testing. They'll shoot live rounds into valleys, and so plants and animals kind of the animals literally retreat, and then the plants die back, and then the ones that are on the sides or in difficult to get to areas are the ones that survive. The rats are a really bad problem too. They eat the seeds, so you don't get regeneration. When you don't get regeneration, then the patriarchs, the older trees, die off, and there's no regeneration in that area for young ones to come up. I hope that answered the question.
Thank you. And then we have
Oh,
one
there was
more.
something about ferns. Let me just say the ferns are really interesting. Yeah, so the ferns do spread really well because of spores, and so they're a lot of times we need birds to distribute our plants, and because birds are dying out, over 100 species in Hawaii, maybe 30 some odd are left right now. So a certain amount are pollinated by birds, but a lot of them are distributed by birds, so they're no longer doing that distribution. And also about the ferns, they spread really well by winds, and sometimes going up into the stratosphere and being blown to different archipelagos. So that's how they spread.
Thank you. So maybe Nina or Ben could answer this one. But Heather was curious, how many species have been named after Ken Wood, and how does naming of species work?
Well, I wouldn't know the exact name. So there's two things in the naming where you see a person's name, well, there could be three different places. So when you name a plant, you can name it after the area it grows in, or a special character like the kuhiheva, for example, that Ken mentioned. Got that name because in Hawaiian it means kind of the unknown or the confused, and there was some difficulty with figuring out it was a new species at first. So you could also name it after a famous botanist. So that happens sometimes. So the plant could have been called Cyania woodii, for example, after Ken. And then there's the person who actually does the naming, so kind of does all the official nitty-gritty work on describing why is this a new species, why could it not be some other species. So you need to, or Ken can explain this in more detail, how you actually do that, because it's his work. So that becomes an author of the species. So at the end of the name, you would see Cyania, kuhiheva, and then, for example, Wood. So that would mean that this was described by Wood. He did the work on that. So there's the authority there, or there could be the person that a plant is named after. But most often it's better to name a plant after something that is a recognizable character, or the area it grows in, or something. So it gives some sort of intuitive sense. But yeah, I don't know if you want to supplement on that, Ben or Ken.
Let's see, am I on? I'm not on mute. Okay. So it's inappropriate for the author of a species to name it after himself. So taxonomists, we never name a plant after ourselves, and it's usually an associate, or maybe even after we're dead and buried in respect. An author will go ahead and name it after that person. Like, for example, I talked about the recent discovery of Melicope stonii from the Mesic forest of Kauai. That was named after Benjamin Stone, who did a monograph of the genus Melicope. And he did so much work on Melicope over the years, but never had a Melicope named after him. So in respect to Benjamin Stone, I named it after him. But a lot of my friends would tease me, and they thought I was naming it Stonii because maybe I liked, I was stoned or something when I found it or whatever. But a little humor there. But no, it was named in respect to Benjamin Stone. And once again, we never name plants after ourselves.
Thank you, Ken. So Lauren, I do see your question. We're going to get to that during the general Q&A, but for now, we're going to pass it over to Ben to talk about technology end of things. So go ahead, Ben, you can take it away.
All right, thanks, Amanda, and thanks, Ken, for all that awesome information. Unfortunately, I had a little mix-up with my presentation. There was a technical issue, so I have sort of a group of other presentations that I've used before. And so I'll start with one that I presented at the World Conservation Congress just this past September, and just disregard the part about Marseille. And it's still a lot of the same stories that I would have been telling with my other presentation. And hopefully I'll have that thing fixed, and we can send out a link to you after the webinar so you can see what my full presentation was going to be. So with that, I will share this. Can you guys see it?
Yes, you're good.
Aloha and good afternoon to everyone in Marseille. My name is Ben Nyberg, and I'm here at the National Tropical Botanical Garden on Kauai. Our organization is focused on the conservation of rare and endangered Hawaiian plants. In the middle of the Pacific, the Hawaiian archipelago has become a case study in long-distance dispersal and diversification. Nearly 90% of the Hawaiian flora is endemic. Unfortunately, human-introduced threats have brought many of these species close to extinction. On Kauai, which I'll be talking about today, there are 250 single-island endemic plants. A third of those species have fewer than 50 individuals remaining. NTBG botanists have been working in rugged and remote terrain for the last 50 years to find and protect these amazing plants. But there are many areas that were too steep or too dangerous to get to. Cliff habitats are refugia for many rare plants. They provide shade, moisture, and most importantly, an advantage against the browsing pressure of feral ungulates. In 2016, we started a drone program to investigate potential methodology for surveying unexplored cliff habitats with flying cameras. Our current drone of choice is the DJI Phantom 4 Pro, a workhorse that balances stability in a variety of weather conditions with portability and image quality. It also has legs which serve as catching assistance in dense vegetation or high wind situations. We aim for a ground sampling distance or pixel size of approximately 1 to 2 millimeters. These high resolution photos allow identification of a range of targets, from large trees to small herbaceous plants. To achieve this resolution, ideal flight distance from the surface is 5 to 10 meters. By nature, these surveys are exploratory, and we're never sure what we might spot. We use indicator species and spotting scopes to guide us in to high-quality areas. Photos are manually classified, and each plant is tabulated. It is a time-consuming process, but extremely effective. Our initial survey results were promising, as we uncovered a number of plant populations in extremely hard-to-reach areas. I'll share a couple of quick stories that illustrate how effective these new methods have been in assessing the distribution and abundance of cliff-dwelling plants. This is Lysa Machias Copulencis. It was down to just 2 known individuals in a single population, and only ever documented in one remote valley of western Kauai. In 2020, as part of a project surveying for another critically endangered plant, we uncovered a group of interesting-looking Lysomachia. We didn't know what it was because it was so far out of range, and few Lysomachia had ever been documented in these dry cliff habitats. My colleagues Adam Williams with the State of Hawaii Division of Forestry and Wildlife and Scott Heinzman with the Plant Extinction Prevention Program rappelled down to the plants to get a closer look. and were surprised to find that the plant keyed out to Lysa Machias Copulencis. As we continued our surveys around the canyon, we were able to locate populations on 4 consecutive ridges. We had found at least 100 individuals. We now have each of these populations secured in seed storage and in our nursery. We observed a number of rare native cliff species, and a potential undescribed species of Euphorbia was also collected. This is Hibiscadelphus woodii, a genus endemic to the Hawaiian Islands that is thought to have adapted to pollination by Hawaiian honeycreepers. It was originally described in 1991 after NTBG botanist Ken Wood located 4 individuals growing on the steep cliffs of Kalalau. Unfortunately, by 2009, all 4 of those plants had died. We thought the species was extinct. In 2019, using drones, we refound it. An exciting discovery, growing in the middle of a 1,000-meter cliff. Drones have helped us gain a better understanding of spatial distribution, habitat requirements, and overall population numbers, and have guided us to significant conservation outcomes. While the extinction crisis is real, we're developing powerful new tools to tip the scale, to explore the unexplored, and to save the rarest of the rare.
All right.
Right.
I have a couple more little things to share, but I just wanted to see if there was any questions from that portion. And then I'll go into a couple of the discoveries that were made this year and some of our next steps.
And we had one come in, CK was asking, so refugia is not a man-made, so like refugia versus refuge in that meaning?
Correct.
Thank you. So I think that's the only one that came in. If you want to go on to your next part.
Okay, this was one that I just made for fun for some of my collaborators with the Plant Extinction Prevention Program. Oops, sorry, that's the wrong video. There we go. And it just has music, and it's them rappelling down to a cliff section on northern Kauai, where we recently found a few groups of Euphorbia Illinois, which is a very rare plant. And we're really excited that we're finding a lot more of it using the drone, and that we've been able to get to it and collect from it. So this has a little music. It was just back in November.
Slow dance with your man. Slow dance with your woman. Everybody slow dance, slow jump. Slow dance with your woman. Slow dance with your man. Slow dance with your woman. Everybody slow dance, slow jump. Slow dance with your woman. Slow dance with your man. Slow dance with your woman. Everybody slow dance, slow jump. Slow dance with your woman. Slow dance with your man. Slow dance with your woman. Everybody slow dance, slow jump.
All right, so that just kind of shows you some of the terrain that we're working in, and how difficult the access is to get to some of these areas. So there's been a lot of these plants like the Hibiscadelphus woodii, for example, that are just in the middle of a cliff system that's too hard to access. And so we were thinking about ways that we might be able to help conserve these species, and remote collection came up as a sort of a pie in the sky idea at first, but the more we talked about it and worked with collaborators, we realized that it might be an effective tool for the future. So we set up a collaboration with this group at the University of Sherbrooke. They have a mechanism called the D-Leaves. And they were focused on collecting vouchers from the tops of trees. But in 2019, we got a grant to customize their mechanism and make it work in vertical environment. So they have a horizontal reaching arm that can move in and work with plants that are growing directly on cliffs. We just had a field test here in October and November, and we're really encouraged by the results that we got. And hopefully early next year, we'll be able to share some of those exciting results. Another thought that we had was remote dispersal. Are there ways that we could put seeds back into the environment from our seed storage? This all came about really because our Limahuli Preserve had some really destructive landslides in an extreme weather event in 2018. And those, we saw a lot of weeds getting into those disturbances, and we're hoping that we had tools to combat some of the weeds getting established in those areas. And so that's where we came up with this. This is our agricultural drone that's designed to spray herbicides and pesticides, but we thought, could we switch out the tanks and the spray nozzles and put native seeds and a hydromulch mix in there to see if we can make this work. So that's another project that we're working on. And, these are just kind of what we see as the next steps of how drone technology can help us out.
Great, thank you, Ben. So we did have some questions come in. So let's answer one or two of them, and then we can do the next portion of the presentation. So let's see, Sherry was wondering, how many drones and pilots do you have? Is there reasonable access to funding to support drone-aided research project? And then a similar question from Lauren came in about how much does one of these drones cost?
Well, the drone that we have chose to use is not a fancy or really complex drone for survey. We're using DJI Phantom 4, which is about $1,500. You can go into Costco and pick one up off the shelves. They're pretty easy to get your hands on if you have a small amount of funding. The larger drones are much more expensive, 10 times as much as that. And we are seeing increased funding to support drone-aided research projects. I think people were a little skeptical at first if it was going to work, but we are seeing that being prioritized. And at NTBG, we have one full-time pilot, that's me, but we also have two other pilots that are trained up in license, as well as one garden director that uses it for photography and videos around the gardens.
Thank you. And then Sherry was also wondering, do you also work with other UAS development research groups, like some of the UAS research centers?
Right now, our only major collaboration is with the University of Sherbrooke, the one that I mentioned. And, we've had some discussions with the University of Hawaii Hilo. They have a spatial lab that does a lot of drone work for rapid ohia death. So there are a few places that we collaborate, but those are the two main ones.
And then Michelle was wondering, do you also use drones for habitat restoration monitoring, like ocular surveys to monitor sapling or seedling establishment, or traditional field visits more effective?
That's part of that project for seed dispersal. So, we were hoping to be able to monitor how well those projects do with the drone. So put the seeds there and then see how they're establishing and, if it's a feasible tool. As far as, what we're doing right now, there's not a ton of habitat restoration modeling done at NTBG. I know that the State of Hawaii Division of Forestry and Wildlife, DOFA, they do a little bit of that with their drones and monitoring some of their outplanting areas.
Yeah, so we have one more. The question I want to go back to was one from Lauren. So Lauren was wondering from Ken, so when rare species are discovered in the wild, NTBG will generally collect and cultivate the species to help prevent its extinction. And the event that it goes extinct in the wild, so is Hibiscus woodii an example, or sorry, Hibiscus delphis woodii, an example of a species that is close to extinction in the wild, but which has not yet been successfully cultivated ex situ? Are there other examples of species that are close to extinction, but do not have the backup population ex situ? And then Vera had a question. She was wondering, when was this Hibiscus delphis woodii rediscovered?
Yeah, so there are examples and quite a few, particularly of species that are presumed extinct. When we say extinct, it isn't necessarily true. It's just that we don't know of any living plants. So there's a qualifier there. It's just that we don't know of any wild individuals. But given that there is suitable habitat, we leave the word possibly in extinction for those. But there are certain ones that were only known from coastal lowland areas, very dry, that we're almost 100% sure that there's cannot be any that have survived. Like, for example, another member of the Hibiscadelphis is the Wildarianus. That one there is extinct in the wild and never been represented in cultivation. And then there's Hibiscadelphis hula Layensis, which is extinct in the wild but is being grown by the public. These days, both Brickhamia are extinct the Brickhamia insignis on Kauai is extinct in the wild. We know of no others, but we've been able to cultivate those. So there are examples of both, and all three: extinct in the wild, extinct totally, extinct in the wild, cultivated. Then we do have ones that are very close to extinction that aren't cultivated yet, and that we're suffering and trying to do, like the Hibiscadelphis woodii. We know of a small little grouping in the middle of those 2,000-foot cliffs. I'm past my prime, and I'm not able to do the rappels that I used to be able to do. But now this new drone system has really opened the doors up. We'll be utilizing the drones to keep an eye on these rediscovery. We've been given another chance to preserve that Hibiscadelphis in Kalalau Valley. But yeah, so it's down to a few, but we've never been able to cultivate it. Sometimes it takes many years for micropropagation labs and horticulturists to find the correct method to get these things established. When I first started working, there was very little known, or that much interest even in funding this type of work. But now it seems like a lot of people are getting on board, and our conservation ethic is growing. So along with the funding, and so I guess that's part of the answer. There's more to talk about. Yeah, there's lots of examples, and I just wish I could show you some pictures of some of these, but I just don't have them ready to pull up right now.
Thank you, Ken. So thanks everybody for all those questions. If there's anything else that pops in your head, feel free to drop that into the chat. We are going to switch to another slide real quick. We have a fun audience participation piece where we're going to share some of the plants that we discovered this year. So let me pull that up for you. All right, there we go. So I don't know, Ben and Ken, if you want to take it away on this one.
I'll let Ben go ahead. I've been talking quite a bit. I can chip in to go ahead, Ben.
You want to trade off? I'll do one and you do one.
Yeah, go ahead and.
Okay. So I mentioned the Euphorbia Illinois already. That one was down to just one small population or a couple, under 50 plants left. And we found it in multiple locations and been able to collect from it, all the way from Honopu up to the North Shore. So I think that was an exciting development in the last year or so.
And just to add, those Euphorbia Illinois, or as the Hawaiians call them, a cocoa, because of the white sap that comes out, the blood-like texture to it. A cocoa means blood in Hawaiian. Those plants are really amazing where they grow there. You remember the videos that Ben was showing of the boys rappelling, the men rappelling, and the edges, on those edges of those knife-edged cliffs, the ridges there is where this plant likes to be. That's why this plant wasn't really found until the 1990s, found and described back then. Anyway, moving on, above that there, Melicope Neale. Now the Melicopes in Hawaii in the citrus family are there's around 50 species, and they're quite interesting. This one here, named after Melicope Neale, who did a book, I think, In Gardens of Hawaii, and was one of the state botanists a number of decades ago. Melicope Neale was originally collected around, I think, 1909 in the center of the canyon of Kauai, around Kahuluamanu, where the Robinsons had a cabin up there, and they would allow researchers to go up there and do explorations. And back then, that species was documented and then wasn't seen again or documented again until 1960. And that was the last time it was seen. And since then, we've never been able to find one until recently, until just a year or so ago. Now this this species is there's only one individual left. We've been monitoring it. It did go into flower and sent out one fruit. This is the hairy exocarp of this fruit. But there was a heavy rain and the seed fell off of that plant. And so we haven't yet, this is another example of a plant that's down to one that we know of, and we haven't yet been able to cultivate it. NTBG has teamed up with the PEPP, the Plant Extinction Prevention Program, and looking at what the next step is. We did try to do 2 air layers, and those weren't successful. Now, all of the PEP folks from the outer islands are joining together and having discussions and trying to come up with a way to cultivate this plant. Interestingly enough, also, I did get funding back in 2015 from the U.S. Fish and Wildlife Service to look for this plant, and I looked quite a bit for it and never found it. So, another lesson here is that, there are still surprises out there. I spent a lot of time looking for it for that for that grant that U.S. Fish and Wildlife gave and never found it. Reported where I'd went and found many other interesting plants, which is also an important point. You know, while we're looking for certain plants, there are other discoveries going on. So anyway, I'm a little bit long-winded on that. Ben, you go to the next one.
Sure. That's a great point about, you know, looking for one thing, but finding another. And I think Lysa Machias Copulencis is a good example of that. You know, I first learned about this plant from Ken when we went looking for Hibiscadelphus woodii, and we visited the area where the last one or two of these plants were. And, you know, I was real interested in it. And as we were flying around in Waimea Canyon, we started seeing something that looked like that. And, you know, now we have maybe 8 populations of this species with hundreds of plants and good seed collections and plants growing in the nursery. So, and that's all in the past, you know, 12 to 18 months that development has been. So I think that's a great example. Back to you, Ken.
Okay, so, you know, Ben mentioned Euphorbia Illinois, and to the left there, there's a little snapshot I took of a press plant, Euphorbia Remieyehana variety, Layensis. Now, the Illinois is just this very tight, compact, 25 centimeter or 10-inch tall shrub that's in these windy zones. This Euphorbia Remieyehana variety, Layensis, gets to be a medium-sized tree of 7 to 8 meters tall. What's really distinctive about it, and we've been looking for it for years, it was only collected once by a guy named Mann and Brigham back in between 1864 and 1865. They didn't have an exact date on it, and never seen again. But as you can see on the capsules that are being shown, it's covered in this white tomentous pubescence. That's a pubescence, not a just plain color, but those are hairs. And all of the Euphorbia Remieyehana varieties are all glabrous except for the Layensis, which is covered in these woolly-like hairs, tomentose hairs. And so, so I was there in the I don't know if you remember the image, there was a big waterfall, Hinalele Falls in the background, and a raging stream, and real big crashing sound. And on the sides of there and going inside there, there was a small colony of these euphorbias, and I was able to make a herbarium voucher. And I did collect seeds at the time, but those were not successfully grown. But we do know where they are. We haven't cultivated it yet, and it's an exciting one. It's especially when you look at the date, only seen one time in 1865. Okay, Ben.
Yeah, that is a super exciting one. I'm love that story. Cyanoacerapolia is Cyania with little heart-shaped leaves, dark green, you know, and was really only known from one area, or known to remain in one area. And on recent surveys in the wet cliffs of Kauai, we were looking for Lysimachia niki and ended up finding, you know, almost doubling the population of this species with a really robust and vigorous population. So I know we're running low on time, so I'll just leave it at that and go back to you, Ken.
Oh, running low on time and back to me. Okay, so Cyania cuyaba there. You know, that's an interesting story. I was with Chipper Wickman at that time. He was the director of Limahuli Gardens. He later became director of our National Tropical Botanical Gardens, and now is the president. So, and his brother John and my longtime field partner Steve Perlman, and we were in Upper Limahuli when this plant was discovered. And there's magic out there too. And Chipper remembers the story. We were talking about this linear leafed Cyania just before this plant was found. I mentioned it, I've been looking for it, I said, for years. And then we got separated because I had these spiked hobbies and I can go up this little waterfall. There was a fallen tree in there and I scurried up and there was a small colony of this linear leafed Cyania in there. And I made a document of it and then came back down the waterfall and they said, well, how was it up there? And I said it was great. And then they go, well, what did you see? And I mentioned this linear leaf Cyania and they all said, nah, no way, because we had just talked about it and it hadn't been seen for over 100 years. And so they're within minutes after talking about this linear leaf thing, then Cyania cuyaba was discovered, and then it went extinct after the '92 hurricane. We had some growing in our nursery, but there was some kind of fungus that got in there, and then they died, and it was all gone, and we were crying. And then just a few years back, we found another population. And so that was like, water on a dry leaf. It was just so refreshing. It gave us another chance. And not only did it give us another chance, we had cooperation with our plant extinction prevention people. We went back to where the colony was and bagged the flowers. And then we went again and collected around 1,000 seeds the first time. And then we went back again the next time it flowered and did another 1,000+ seeds. So we really nailed it right this time. Even though there's a lot of sad stories, there are some really joyous ones, and Cyania cuyaba is one of those.
All right, thank you. That was really fun hearing all those stories from you all. So I know that we're over time. Real quick, I just want to launch a poll. So out of these 6 discoveries that we made in 2021, which one is your favorite? So go ahead and take a moment, and if you'd like to answer that poll for us, I'll share the results. And Ken and Ben, if you have time, maybe 5 more minutes, maybe we can get to a couple of those other questions that came in. And feel free, I will be sending out the recording to everybody who registered. So if you need to leave, go for it and you'll get the recording. But if you want to stick around and as we answer a few more questions, you're welcome to that as well.
So Ken, I don't know if you see this question from Sherry. Are you guys using GIS to document plant locations and to help stratify areas for priority exploration? Or is it more the deep personal knowledge of your group that guides the selection of areas?
So yeah, so let's see. Are we using GIS to document where our plants are? Is that part of it?
Yeah, or are we more using, knowledge of the ecosystems to guide us? And I think the answer is we use both. We use both those tools to help us. But I was interested to hear what your take on that was.
Yeah, so initially when I first started working deeply in trying to find, it's like an Easter egg hunt, going out there and finding the Easter egg on Easter morning. I spent a lot of time at Bishop Museum, which is the state museum where a lot of the historical collectors deposited their herbarium specimens, and I had a list of 100 or 200 species that there wasn't much known about it. I studied where their ecological niches were thought to be, and then I applied my familiarity with the high islands of Hawaii to look for those plants in particular. Mostly they're usually single island endemics. In other words, they only occur on a single island, and that's pretty much why they're quite rare. So, that was one tool, is to look at where our 4 collectors had gone and found it, and utilizing that knowledge, and to know, if that area is already destroyed, to know where similar habitats are, to go to those sites. Another thing that happens when you spend a lot of time within certain ecosystems, whether they're common places like on the street or in deep forests, after repetition, the subconscious and the mind becomes familiar with what's around you. So you're not thinking that much about things that normal to everyday things. You're kind of looking and you're moving through, and you get to see this repetition of what type of species occur as you're working through. And sometimes just before you see another rare species, it'll even come to your mind because the last time you saw it, it was in a similar condition. And so then you're on the alert to be aware that this plant could be in that area. And I tell you, there must be hundreds of times when that would come. I think of a species and then I'd look a little bit harder and it would be there. So there might even be, you know, besides the documentation that was done in the past, there might even be some subconscious awareness that bubbles up when you drop your mind and you're just present. That gives you a little bit of an edge on being able to find some of these species.
Yeah, my background's in GIS, so I think that's kind of what I go for first. But I have the luxury of working with Ken, who's a maestro of the Hawaiian ecosystems. And I think it is partly a developed understanding of what is where and what good habitat looks like that Ken mentioned. Because when I'm flying drone, like you said, I kind of drop out of thinking about other things and more are just looking at species composition and a lot of other things that kind of help guide the drone into the right place.
Thank you. So I just want to, real quick before we check back on questions, share the poll results with you all. So overwhelmingly here, we have that Cyania. So thank you all for participating in our little poll that we had here. Appreciate it. So let's see. Let's try to
just
get.
to say, and I'm sorry to interrupt, but yeah, that is a good choice because actually, you know, it was first found in Limahuli Garden, which is, you know, 1,000 acres on the North Shore as part of our garden. So I think that is a really cool choice, and what a wonderful family the lobeliads are.
Oh no, thank you, Ken, for interrupting and sharing a little. Appreciate it. So let's try to get maybe one or two more questions, and then if we didn't get to something, we'll try to follow up with you via email. So Sherry was wondering, let's see, AI wishes it could model your awareness and what you have incorporated intellectually. I'm not sure, maybe that wasn't a question, but
Let's see, I'm not on mute, yeah. So yeah, as far as we are training, over the last 30 to 40 years, I've had a lot of postdocs that come through the garden, and I've worked with in the field, and a lot of these lessons we learn together, I can share a lot of my experience, and then I'm learning a lot from the young folks too on technology, and so there's you know, awareness is a certain you know, it's definitely, if you can kind of close off the dialogue of likes and dislikes and ups and downs and being present and focusing on a scientific subject, it just keeps expanding and expanding. And yes, we do keep on learning more and more, and we're able to apply it. And what's really beautiful about it is it isn't just theoretical applications that we're doing, but we're actually existentially gathering seeds that hold the mystery of life for these beings that have survived longer than probably Homo sapien has been around. And so, you know, coupling the conservation science and awareness at a time when it really is a science of, it's a stressful science because there's a deadline to, you know, we're seeing things die off so quickly and ecosystem change. So, you know, the more we can share our awareness with others, the better it is. And so that's one thing really good about NTBG is that we always have students coming through. It's not only a conservation organization, but we have grounds where we can grow these things out. We have an herbarium where we can have people come and study them. And then we have seeds being stored by professionals here. And then we also have a DNA lab.
Hi, thank you. So I do think we got to all of the questions. Thanks to those of you who stuck around. We appreciate it. And we just want to thank you so much for everybody who attended one of our fall webinar sessions, or maybe even more than one of our fall webinar sessions. I appreciate your participation, and we're really looking forward to doing this again in the spring. So we will be sharing this recording out. So I will pass it on to you all sometime this afternoon. And I'll also include a survey. So if you wouldn't mind, if you haven't already, filling out our quick survey for us to let us know how we did, and that'll help us improve our planning for our spring session. And then I also just wanted to share an article with you. I'll drop that into the chat now. We are in the middle of our fall appeal. And this year, we're really focusing on a line from our original charter that it talks about how the need is upon us. And this is over 50 years ago, and it still, rings true today. And one of our latest stories that we're telling right now, the one that I just dropped in, is about the founding of NTBG and about these adventurous botanists like Ken's predecessors who, went out and did this work even back in the early 1900s and in the middle of the century as well. So if you're inspired, please feel free to visit our support page to find ways to support us and help us continue the work like what Ken and Ben do. Without further ado, we'll go ahead and end the session. And again, we really appreciate it and hope to see you in the spring. Thanks, everybody.
Thank you, everybody. Aloha.